Welcome to the PubMed Daily Review for April 21st, 2026. Today we scanned 3,304 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, precise, specific gene editing via a compact GoCas12m, FokI chimeric nuclease by Tin Marsic and Magdia Mahfouz. Let's break down its key contributions. CRISPR gene editing has revolutionized our ability to modify DNA, opening doors for functional genomics and potential therapies. However, current technologies often struggle with delivery challenges and unwanted off-target edits. Many CRISPR systems are simply too large to fit into common delivery vehicles like AAVs, limiting their therapeutic reach. This study explores a novel approach to overcome these limitations. To address these challenges, researchers developed a novel gene editing platform by fusing a compact, catalytically inactive Cas12m protein with a FokI nuclease. This GoCas12m FokI system utilizes a dual-guide RNA strategy to bring two nuclease units together, enabling precise DNA cleavage. The study tested this system in human cell lines and on several endogenous human genes, evaluating its editing efficiency and specificity. The results were highly promising. The GoCas12m FokI editor showed robust and efficient editing at multiple human gene targets, including HBB and AIFM1. Crucially, extensive analysis confirmed no detectable off-target edits, indicating a high degree of precision. Furthermore, the engineered editor is significantly smaller than conventional systems, making it compatible with AAV vectors for improved delivery. This study presents a significant advancement in genome editing technology. The GoCas12m FokI system's compact size and remarkable specificity make it a powerful tool for therapeutic applications, particularly in gene therapy, where efficient delivery is paramount. While these findings are exciting, further research will be essential to fully explore its potential and ensure its safety and efficacy in clinical settings. Up next, our second paper out of 5. Proline restores mitochondrial function and reverses aging hallmarks in senescent cells by Debanik Chowdhury and Stelios T. Andreas from Department of Chemical and Biological Engineering, University at Buffalo, New York, 14260, USA. Let's break down its key contributions. Cellular aging, or senescence, is often accompanied by a significant decline in mitochondrial function. This dysfunction is thought to be a major driver of the aging process. Our previous research indicated that a specific protein, NANOG, could help reverse this mitochondrial decline in senescent cells. This led us to investigate whether other interventions could achieve similar rejuvenating effects. To explore this, we used senescent mesenchymal stem cells as our model system. We treated these cells with Proline and then employed various techniques to assess its effects. This included looking at gene expression, specifically for enzymes involved in proline synthesis, and directly measuring mitochondrial health and turnover. We also evaluated several other established markers of cellular aging to see if Proline could reverse them. Our results were quite promising. We found that supplementing with Proline significantly alleviated the severe mitochondrial respiratory impairment observed in senescent cells. This improvement was linked to proline-inducing mitophagy, a process of clearing damaged mitochondria, through the activation of AMP and upregulation of Parkin. Furthermore, Proline treatment successfully reversed other key aging markers, including DNA damage, senescence-associated beta-galactosidase activity, and the expression of inflammatory cytokines. In conclusion, this study highlights Proline not just as a building block for proteins, but as a crucial metabolite capable of rejuvenating senescent cells. By promoting the clearance of damaged mitochondria, Proline offers a potential new strategy for combating age-related decline. These findings open exciting avenues for future research into proline-based therapies for various age-associated conditions. Up next, our third paper out of 5, Pharmacokinetics, Pharmacodynamics, Efficacy, and Drug-Resistant Selection of Injectable Long-Acting Lenacapavir Pre-Exposure Prophylaxis, PrEP, against HIV by Hyong Kim and Max von Kleist from Project Group 5 Systems Medicine of Infectious Disease, Robert Koch Institute, Berlin, Germany. Let's break down its key contributions. Daily oral PrEP has been a game-changer for HIV prevention, but sticking to a daily pill can be tough for many. Long-acting injectable options like Lenacapavir, given twice a year, offer a promising alternative. However, we need a better understanding of exactly how well these injections protect against HIV and what risks, if any, emerge, especially after someone stops taking the medication. To tackle these questions, the researchers developed a sophisticated mathematical model. This model integrates Lenacapavir's pharmacokinetics, how the drug moves through the body, with its pharmacodynamics, how it affects the virus. They also factored in variability between individuals and simulated the emergence of drug-resistant strains, providing a comprehensive view of Lenacapavir's performance. The model predicted that a concentration of over 5 nanograms per milliliter is needed for full protection against HIV, a level maintained for over 50 weeks after the last injection in an average individual. However, the study also found that for certain Lenacapavir-resistant mutations, the window for resistance to emerge overlapped with Lenacapavir's steady-state concentrations, highlighting a potential concern. This study provides valuable insights into Lenacapavir's effectiveness as an injectable PrEP. It reinforces its potential as a long-acting option while crucially highlighting the need for careful planning around discontinuation to prevent drug resistance. Future work should focus on optimizing dosing schedules and developing clear guidelines for stopping Lenacapavir safely. Up next, our fourth paper out of 5, AICellType, a large language model-based platform for accurate cell type annotation by Chuxing Chung and Mailin Jin. Let's break down its key contributions. Single-cell and spatial transcriptomics have revolutionized our understanding of cellular diversity. However, a major bottleneck remains accurate cell type annotation. Traditional methods rely on fixed gene markers, which can be limiting, and manual annotation is time-consuming and subjective. Recently, large language models, or LLMs, have shown potential in interpreting biological data, offering a new approach to tackle this challenge. To find the best LLM for cell type annotation, the researchers systematically evaluated 79 different models. They used over 1,000 single-cell and spatial transcriptomics datasets, comparing LLM predictions against expert annotations. The evaluation framework focused on biological relevance and annotation robustness. The key finding was that Claude 3.5 Sonnet emerged as the top performer, balancing accuracy, speed, and cost-effectiveness. The study found that Claude 3.5 Sonnet achieved a weighted accuracy of 76%, significantly outperforming models like GPT-4. It also demonstrated impressive cost efficiency at just 41 cents per 1,000 cells and a rapid median annotation speed of 3.7 seconds. Based on these results, the team developed AICellType, a user-friendly package and web platform that leverages this powerful LLM for accurate and scalable cell type annotation. AICellType represents a significant advancement, providing a robust, efficient, and accessible tool for cell type annotation that overcomes many limitations of previous methods. By harnessing the power of LLMs, it democratizes advanced analysis for researchers. The platform's open-source nature and flexibility allow for future integration of new models, paving the way for even more powerful and specialized tools in biological research. Up next, our fifth paper out of 5. AngioPep, 2 decorated bacterial outer membrane vesicles penetrate the blood-brain barrier for glioblastoma chemo-immunotherapy by Pierre Tariq Shah and Chengjun Wu from Acor Laboratory, Qingdao Central Hospital, School of Health and Life Sciences, University of Health and Rehabilitation Sciences, Qingdao, Shandong Province, China. Let's break down its key contributions. Glioblastoma remains a devastating brain cancer, largely due to the formidable blood-brain barrier that prevents most drugs from reaching the tumor. Compounding this, the tumor environment itself suppresses the immune system, and cancer cells often develop resistance to treatment. This means even with aggressive therapies, patient outcomes are often grim. We need innovative approaches to effectively deliver therapies past the BBB and engage the immune system to fight this disease. To tackle glioblastoma, we engineered bacterial outer membrane vesicles, or OMVs. Using a genetic engineering approach, we decorated these OMVs with a peptide called AngioPep 2. This peptide is designed to specifically target receptors on the blood-brain barrier, allowing the OMVs to cross it. We then loaded these engineered OMVs with the chemotherapy drug doxorubicin. We tested their ability to penetrate an in vitro blood-brain barrier model and confirmed their brain accumulation in live mice using imaging. In our glioblastoma mouse models, the doxorubicin-loaded OMVNG demonstrated remarkable efficacy. We observed significantly enhanced penetration across the blood-brain barrier compared to OMVs without angiopep-2. Crucially, systemic administration of DOXOMVNG led to a significant extension of survival time in mice with brain tumors. Furthermore, this treatment induced a potent anti-tumor immune response, characterized by elevated IFN-gamma levels and recruitment of immune cells to the tumor site. This study highlights a promising new approach for treating brain cancers like glioblastoma. By engineering bacterial outer membrane vesicles to carry therapeutic payloads and target the blood-brain barrier, we've shown a viable method for delivering drugs directly to the brain. This platform not only delivers chemotherapy effectively, but also harnesses the immune system to fight the tumor. These findings suggest that this engineered OMV system could be a versatile tool for developing future therapies for a range of central nervous system diseases. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.